have a multiple task speakers over there, and I would like to announce just 2 new things we're gonna have there. On the very first day, we're gonna have some companies that are going to be looking for students to work them, and we're gonna have the Brazilian Service for Enterprise that will be held the Ideathon. Ideathon is it's like a hackathon, but a hackathon for new ideas for those companies. And we're going to be there on the very first day. Aside, we're gonna have a work-on seminar with the guys from Elixir Europe to make your data more fair. So I would like to invite all of you to be on the X meeting. And on Thursday, we will have now the most traditional part of the X meeting is the X party. It's going to be very great. Yeah, it's going to be a very great one. Unfortunately, due to forecast predictions, we have to call out the X Arraiá, our Festa Junina. Will be able to do it. So we shift to another brand. It's a X party. But the fun and the connections and the dance, we're going to be the same. Don't worry.
Okay.
And one last thing. Yeah. Everyone who will be on the trend topics might be able to win Amazon Echo. So don't forget to go at the trend topics talks during the X meeting, and you might leave with one Alexa from there.
Yeah.
That's it.
Thanks for the infos. It's kind of too late to invite Derrick to come to this one, but what about the next one, Derrick? Do you know Brazil? Have you been to Brazil yet?
I have not, no.
See?
So, in name of the Brazilian Association of Bioinformatics and Computational Biology, you are formally invited for the next year X meeting.
Yeah, that's great.
For a talk, for a keynote, for a course, you are now invited. It will be a pleasure. It will be a pleasure to have you here. Don't worry, next year we'll find another party, but the X meeting is going to be great as this one.
Yeah. What about that, Derrick? Are you into this to come to talk in person?
Perhaps. I have to see how we can.
We can
make that work. Right?
We don't even have the place defined yet, but maybe by the beach, maybe. Yeah.
Yes, maybe by the beach. It's a surprise.
Surprise. Okay. Well, as I told you, we go like popcorn, and popcorn are approaching to a good situation here. Let me check the YouTube. 5 seconds after, we have 20 people there already. We have some people here. So 39, one more popping. We'd like to introduce Derrick. Marcelo, did you?
Yes, I can.
Marcelo, yeah, you can? Sure.
Yeah.
All right. And Derrick, you please say or some more details of your today's work, etc. So let's go for the 40 people, still popping people. Sometimes we start 10 minutes after 2 o'clock, but I think we can go ahead. And okay, Marcelo, you can lead. Derrick, thank you very much for coming and talking to us about Kraken, so important programming software for these people here. And I hope you have time after for their questions. And so thanks a lot. And I will pass the word to Marcelo, and then we can get started.
Okay,
thank you.
Thank you very much, Miguel. It's a pleasure for us to present Dr. Derrick Wood. And he he works mainly in developing algorithms for software to solve problems that deal with DNA sequence data. And he's most interesting on microbiome genomics and microbiome analysis. He has his PhD degree from the University of Maryland on the computer science department, and then moved to Johns Hopkins to work with Ben Langley. His most known contribution is the Edge Pro, and the Kraken is who doesn't know, is one of the fastest metagenomics sequence classifications using exact reads. Thank you very much, Derrick, to accept this invitation to talk to us today. And please feel free to continue and enhance your small presentation that I have done for you. Thank you very much. Derrick, the words are for you now.
All right. Thank you, Marcelo and Miguel. Yeah, as was mentioned, you know, a lot of my focus has been on things like the microbiome and metagenomics, specifically around a program like Kraken. I've also spent a good bit of time also working around cancer genomics. Not going to be talking too much about that here today, but just various things around there in terms of helping to improve diagnostics and make them more accurate and more efficient. Much of what I'm going to be talking about today is, of course, Kraken. That's the talk is titled "Releasing the Kraken," and I think we can just get into it. I will try to pause a few times where I think it makes sense, but certainly if people have questions, please just let me know along the way. Okay, hoping everybody can see that.
Yes.
Okay, great. So what I'm going to be going over first is just the basic problems of microbiome sequencing and things that we might want to do with a particular sample that we have sequenced. There are a number of various questions that you could ask. The top three that I've listed here are kind of the ones that I think are pretty relevant and came to my mind. You know, the first being, okay, which organisms are actually in this sample that you've gotten from somebody's microbiome? You want to know what species are there. You'd also like to know how much of each one is there. Is it dominated by a particular species or genus? And finally, you want to get these answers somewhat quickly. If you were to have to wait several weeks in order to get an entire sample processed, that's less than ideal, and it slows down either your research or your diagnostics. And so getting answers quickly is going to be an important thing. In the talk today, I'm going to really focus on answers to two of these questions, namely identifying the organisms in the sample. This is going to be the problem of taxonomic sequence classification. You may have also, if you've read a number of papers on this, some people also call this binning. And the question of how to answer these things quickly, and we'll talk about the various techniques used specifically in Kraken to classify the sequence data very quickly. The second question is a very valid one in terms of figuring out exactly how many copies of each genome are present in a sample, but it's actually a much more complex one than you might think. And so it's a little bit beyond the scope of what I've got time to talk about today. So we're just going to talk about mainly these two things. And then I'll also talk a little bit at the end about various applications and extensions to use of Kraken that others have developed that I'm familiar with and want to bring to your attention. So the general problem of trying to classify these various sequences is that, when you sequence your DNA from a microbiome project, you're going to be just grabbing data from a particular host environment. This could be, a person's skin, it could be their gut, but there's an environment that you're looking at, and you're not doing any selection for particular organisms that you know of. You are actually trying to get a wide profile of what's there. And so in this example here, we've just got a number of organisms represented by the various colored dots, and the sequences that we would get from DNA sequencing colored in the way that corresponds to the species that they came from. Now, of course, in practice, though, when you sequence DNA, you're not going to have these species labels present. You're not going to have any sort of label present. And so what we need to do is figure out a way to get some sort of reliable label associated with each of these sequences, so that we can then do some sort of downstream analysis that will be of interest to us. And the labels that we're probably going to be using are those associated with the taxonomy. What we've got here is an abbreviated version of the taxonomic tree supplied by NCBI. And the sequence at the bottom here is actually from a particular strain of Helicobacter pylori, the 26695 strain. And if we were to try to assign a taxonomic label to this sequence, it turns out any of the nodes, be it H. pylori 26695 or any ancestor of that, would be a correct label, technically speaking. Of course, as we know, the people who are working with this kind of data are going to want a taxonomic label that's much closer to the bottom of the tree. Knowing that you have data that is from bacteria or even a particular phylum may not be as useful as knowing it's a particular species or a particular strain, which can give you some idea of the pathogenicity of what you're dealing with. So the closer we can get to the bottom of the taxonomic tree in our labels, the better off people are going to be, and the more use they're going to get out of the method to perform this classification. Now, one way that was used in the past to actually assign these labels was doing something like, say, running BLAST, and using the best hit that you might get from running BLAST. So the fastest BLAST program out there is actually MegaBLAST. There are a number of other flavors of BLAST, but this one tends to be the fastest. And in this case here, we see three reasonably good alignments to various genomes. Of course, one being a strain, the 26695 strain of H. pylori. We also have a second strain of H. pylori that is a little bit worse in terms of the alignment, but it does align pretty well with a very low expectation value. And another species within the Helicobacter genus that is also a very low E-value. So this kind of best blast hit method would probably assign this the H. pylori 26695. The main problem with using this, even though fundamentally it makes sense to do an alignment to see where the closest match is between the sequence that came off the machine and your reference database, is that it's a very slow thing. As we get to sequencing tens of millions or even 100 million or more reads per sample, processing only 4,000 to 7,000 of reads per minute is just going to take an enormous amount of time, and you're going to really be limited in what you can do with the results, because they'll just take forever to actually obtain. And this, part of the reason why this takes so long is that this process of traditional alignment is that it's actually trying to give you information about the shape of the alignment and the specific location within your reference where the read sequence best matches. So that's a very involved question. It requires the solving of a dynamic programming problem, and it just takes a good bit of computational work in order to come up with the answer. But in terms of the taxonomic classification question, what we're really interested most in is just, well, which genome did this particular read come from? And we don't necessarily need to know what particular sequence variants might exist or indels, or where in the reference a particular read came from. We just need to know which organism it came from. So this isn't to denigrate those who might want to get this information and use it for some other downstream analysis, but if you are just interested in getting the the specific answer of which taxonomic node makes the most sense for that sequence, this is more work than actually needs to be done. So instead, what we came up with back around, I think it was actually in late 2012, was the idea of building a program that was specifically just for taxonomic classification. Rather than relying on traditional alignments, we're using a more simplified version of it that is doing exact alignment. So we would take a read, divide it up into all the different overlapping k-mers that might exist. In this case, we're using subsequences of 31 base pairs long. That's the default in Kraken 1. And the alignment here is an exact alignment between that k-mer and our reference database. And the idea here is simply, if you've got an exact match that's that long, it's very unlikely that the read would not actually come from that particular genome. So this just relies on a very similar idea to using traditional alignment to get the origin of a particular read, but doing a lot less computational work to get there. So to do all of this work, Kraken requires a few different things. One is actually just a set of reference genomes. This is usually somewhere on the order of several thousand to tens of thousands of different bacterial genomes in practice. A taxonomy. NCBI will generally give this to you, but some people will create custom ones. And then for every one of the reference genomes that you've got to tell Kraken where within the taxonomic tree that genome actually maps to. From this information, Kraken will build this very sizable dictionary that maps every different k-mer that's found within your reference set to what's called the lowest common ancestor of every genome in the reference set that contains that k-mer. And I'll give a few examples of what that looks like in a moment. In addition, there's an index that was created in at least Kraken 1 that will allow this to be dictionary to be queried quickly and keeps us from having to store any extra information in the dictionary than just this mapping of k-mer and lowest common ancestor, or LCA, information. So the idea of the LCA, or the lowest common ancestor, is that for a given k-mer, which will be represented by the various k's that we have here, we only want to store one value inside of our dictionary because there could be k-mers that are very common across many different genomes. And if we had to store a very large amount of taxonomic information for some of these k-mers, it could actually cause the dictionary to become very large in size and a bit unwieldy. So we've got within the framework of Kraken a constant storage of one taxonomic ID for every k-mer. And in this case, the orange k-mer that we see here is found in both, let's say, Salmonella enterica and Yersinia pestis. So within our dictionary, we're going to store the lowest common ancestor of those two nodes, which is at the family level, the Enterobacteriaceae. Now this, let's say, blue k-mer, which is found in Salmonella enterica and H. pylori, because these two species are actually in different phyla, that turns out then that the LCA for that particular k-mer is actually going to be at the domain or kingdom level, which would be bacteria. So this particular k-mer, because it is spread across such a wide area of the taxonomic tree, ends up being not quite as useful for classification. That said, Kraken will still try to use it and try to use all the supporting evidence that it can to perform its classification. So this is the general algorithm that gets used by Kraken to classify any particular read or query sequence. It's first decomposed into its various overlapping k-mers. Each one of these is then queried against a pre-computed database. So this is all done ahead of time. And we see 4 different taxonomic IDs in this example that correspond to different nodes in the taxonomy tree. And in this case, we have one sort of very vague and not as helpful LCA here, and a lot of k-mers that are associated with a little bit more specific node, and then 4 of these k-mers that are a little bit deeper in the tree. And then we've got one node that's well off from all the others that just came in at the end of the read. And often in a situation where this might happen, this is often an error, due to sequencing. And the classification algorithm is designed to try to add some resistance to this sort of thing appearing in the middle of a read, or even at the end of the read. And so the way we do that is by taking the taxonomy tree and only looking at those taxes that are actually hit by the query sequence here. So we have a much smaller tree that we'll examine, and the weights associated with each node here are simply the number of k-mers that were found in the query sequence. So we have 1 for purple, 10 blue ones, 4 orange, and 1 red. And what we'll then do is go from the leaves of this particular classification tree, and we can run up to the root in each of them, and we will get a sum for each particular leaf. So in this case, this one would have a sum of 15 because there are 15 along this yellow path, whereas the red node would have a sum of 2. And so in this case, the evidence for the orange classification far outweighs that for the red one, and we would use the orange node as the label that Kraken will give this particular sequence. So just to give some idea of how Kraken 1 performed, at least around this data. So this was once again back in 2014. So this is about 10 years ago, but the programs that were around at the time, the naive Bayes classifier, Fenbel, even a confidence filtered version of it. Kraken had an accuracy, positive predictive value that was considerably higher than those, and a sensitivity that was well on par with them. But most importantly, Kraken ended up being considerably faster than any of the other programs that we were comparing against. And one thing of note is this program Metaflan, which some of you may be familiar with, was largely introduced in part because some of these other programs were just quite slow. And so what Metaflan does is tries to classify a small fraction of the reads that are associated with marker genes, and therefore it can do its work reasonably fast. I believe it still uses a bow tie to do its alignments, but it is only going to be classifying a small fraction of the read. So you'd have a sensitivity somewhere around, I believe, 10% or so, perhaps even less. So it's for this reason that we show Metaflan as part of a speed comparison, but not an accuracy comparison on this chart. So we wanted to talk briefly about how Kraken 1's structures actually work. Part of the algorithm behind this is to use the concept of minimizers. For any particular k-mer that we might want to work with, say we have a 31-mer, we also have a specific minimizer that can be defined by looking for what we call the minimum Elmer within that k-mer. The example that we've got here is that if your L is 2 and K is 5, what we can do is look at this 5-mer, break it into substrings of length 2, and then just do a lexicographical sort of these 4 possible Elmers that are present within the k-mer, and that gets you this AT string as the minimizer for that particular string. In practice, you also look at the reverse complement of this as well, and so what this ends up doing is for any particular k-mer, there is one minimizer that is defined for that particular k-mer, and the real benefit of this is that when we have overlapping k-mers, because they've got so much sequence that overlaps, there tends to be this property where these overlapping k-mers will share the same minimizer. And that means that as we are going down our reads from left to right and looking at the various k-mers, we will be querying a k-mer that often shares the same minimizer as the k-mer that we just queried. So this is one of the things about Kraken 1 structure that we did to keep things moving very quickly, is that the data itself is organized in such a way that uses the CPU cache, so that one k-mer query is often accessing the area that was just accessed by the past k-mer query. And I'll talk in the next few slides about how that is actually designed. But it ends up speeding up Kraken significantly, as opposed to us not doing that and just doing a simple binary search over entirety of the list. So the particular structure that we've got here is this sort of sorted list of k-mers and taxonomy IDs. And the k-mers are sorted first by their minimizer. So all of these records here are different k-mers that have the same minimizer, as represented by the orange bar here. And then after that is another minimizer represented by the blue bar, and then some other one that is not of interest here. But for looking up a particular k-mer in this list, we've built an offset array that tells us after we compute the minimizer, we can just look within this array and the minimizer corresponds directly to a specific number because we can store all of these values as integers rather than as strings. And so we can look it up very quickly, finding one minimizer's offset within the large table, as well as the subsequent minimizer that might exist. And then we've got a defined range within that list that we can do a binary search on. And this particular area here tends to be much smaller in practice than doing the entire sorted list. And so because of that, the binary search is considerably faster, and it's also going over the same area for usually about 7 or 8 k-mers in a row. One particular downside of Kraken that I think a number of people noticed offhand once it came out was that although it ran very fast, it certainly did use a considerable amount of memory. Even back in 2014, the default dictionary that we would use combined with the index required over 70 gigabytes of memory in order to run efficiently. And every new distinct k-mer that somebody might want to add into their reference or that got sequenced and deposited in the RefSafe database ends up causing this to grow. And so as a result, we ended up seeing a number of sort of competitor programs that were trying to help solve this problem by becoming more memory efficient and trading a little bit of speed in the process. So we see a couple of examples here of the Clark and centrifuge programs are saving some memory, but losing a little bit of speed in the process. And it was this that led me to want to see if we could also get kind of very similar reductions in memory usage, but without sacrificing the speed. And so that's where we came in with Kraken 2. And this was released in 2018, and we published it in late 2019. And as we can see here in the plot, Kraken 2 is about 5 times faster than Kraken 1, while using about 15% of the memory that Kraken 1 did. So it brings it, right on par with something like a centrifuge. Not quite as memory efficient as that, but still definitely on that level as opposed to where it was before. In terms of the accuracy in our strain exclusion testing, the results look very similar across the board. Most of these programs, in terms of exactly how they're going to perform, tend to do something within the very same ballpark. We do definitely see a small increase in viral accuracy for Kraken 2 over the other programs, and that in some ways is due to some of the technical details of how Kraken 2 works, which I'll be going over in the next few slides. But primarily the memory benefits we got were by switching from the sorted index list that we talked about to this new compact hash table, and also subsampling what we actually put into the hash table that we're looking at. So rather than putting in every k-mer that might be in the reference set, we're only putting in minimizers. There's also a method for hash-based subsampling within Kraken 2, which will allow you to ensure that the size of the database is within a set limit that you specify, and then we use just a hash function to ensure that will work. Part of the speed gains that we've got were largely due to improved thread scaling based off of some work that happened in Ben Langmead's lab in terms of how we process the individual read data and actually parse it out. We've also got the ability to do a translated search where we will take the nucleotide sequence of the reads and translate it into all 6 possible protein translations and compare against a reference database of proteins. And as we can see with the viral accuracy there, that also, for both programs that we've got the translated search data for, tends to provide an even greater increase in viral accuracy. So if you were focused on doing viral metagenomics, that could be something of real importance to you. And exactly how this ends up working, if you've seen this before, I am going to go into a bit of detail on it. So the compact hash table we got is a probabilistic data structure. And what that means is that the table itself has a small false positive rate associated with it. So there is the possibility that if you query it, you can, it'll tell you that, oh yes, I actually have seen that gamer, when in practice it actually did not. But the probability of that happening is well under 1%. We've also got requirements within Kraken 2's code to ensure that that multiple minimizers trigger a call rather than a single one. So the actual false positive rate that comes out of Kraken 2, as opposed to just its hash table, is very, very low. I believe it's probably somewhere under 0.01% at this point. But part of the main reason we switched to this is that it gives us records that are only a third of the size of what we had in Kraken 1. So with Kraken 1 and the record storage that we see here, we had to store the full Kamer. So all 64 bits of that had to be used, as well as 32 bits for the LCA. And here we do a good bit of work to try to reduce exactly how much we need to store in order to get our memory down considerably. So the way we do that is, for a particular Kamer, we'll first compute the minimizer, and we have a space seed mask, which also helps just to improve accuracy. But this goes through a hash function, which of course will spit out a kind of seemingly random hash code. And we take the most significant bits of that hash code and use that as the thing that we actually store in our hash table. So these 15 bits here from the beginning are what gets stored in the table, and that's what gets searched for if somebody else comes along later with a new Kamer. We would go through this process of getting the minimizer and hashing it and looking at the first 15 most significant bits. So that combined with using the least significant bits of the hash code to give us a position within the table to start searching is how we then work to actually perform a sort of linear probing search that we would do as any sort of normal regular hash table that uses linear probing does. And so it's through these measures here that we're able to only store about 15 bits for each particular Kamer and get away with only needing a third the size for a particular record. In practice, the hash tables that we use, especially because of this false positive issue that could exist, and because we're using linear probing, we have to include some sort of slack space. I think by default, the load factor ends up being tuned to 70% for Kraken 2. You can adjust this as a user build parameter. If you really want a much lower false positive rate, you can incur the extra memory usage. You could bring that load factor down to something like 20 or 30%. And not only would that reduce the false positive rate that the structure would give, but it also would actually make just the queries go a little bit faster because there's less actual probing going on. The minimizer subsampling point here is a way that we also limit exactly how much we're storing. Rather than storing every k-mer that might exist, or every 31 mer as we did in Kraken 1, we compute the minimizers of our k-mers, and it's that governs what we actually store. And by using a K of 35 and an L of 31 with Kraken 2, this results in us storing only a third of the data that we would have actually stored. So this cuts down on the amount of data that we need to keep around, but it also ends up actually boosting sensitivity slightly for these k-mers that might be near the reference but aren't an exact match to it. As does the space seed stuff that we're using here. So these two things here would bring us down to a ninth of the size of a Kraken 1 database, but the slack that I talked about with the hash table is why we're actually up around 15% instead of 11% of a Kraken 1 database size. And then finally, for the translated search, we use a reduced amino acid alphabet there, rather than storing all 20 different amino acids that what somebody might use, as well as the 21st value for a stop codon. We've reduced that down to 15 amino acids in a stop codon so that we can fit each amino acid within 4 bits of information, so that we can have longer amino acid k-mers and minimizers in our storage there. So I want to, as we finish up, just talk about a few different programs that have used Kraken's results and things that somebody might do, if they're designing a pipeline that uses Kraken. One of these is the Kraken program. Now, if you recall, the Kraken can assign a taxonomic label to a read that is not necessarily down at the species or strain level, but might be a bit higher up. And when we have that situation, for Kraken is only looking at each read in isolation, but Kraken will actually take all of the Kraken results for a particular sample and extrapolate from that where these reads that are assigned to higher taxonomic levels, where they might actually accurately be placed if you were to try to drive them lower into the tree. So we see some examples of where that happened here, especially this particular example with the Serratia here. A lot of the read data there was classified at the genus level, and Kraken was able to say, well, at least the counts for those can be assigned to this particular species. And Kraken was initially created for Kraken 1, largely by Jin Liu, but we were able to apply it to Kraken 2's results as well. And it turned out to actually reduce the error using the Kraken 2 results. So we were pleased to see that there. Additionally, the Kraken Unique program was a program developed as an extension of Kraken 1 to solve the question of presence or absence of a particular species. And this was largely brought into play because a kind of a known problem is that there will be certain sequences in our reference databases that are, let's just say, erroneous. Either they're assigned to the wrong taxa, or they have some sort of sequencing error or assembly contamination present. And that little bit of, let's say, assembly contamination could result in a large number of reads in a new sample being assigned to that erroneous taxa. And one way we can detect that is if we were to look at the particular k-mers that actually result in classification. It turns out that for that erroneous taxa, we'll often see a situation where it's only a handful of distinct k-mers that are actually causing several hundred or even several thousand reads to be classified to a particular taxon. And that goes against our general expectation of how k-mers should be distributed across a reference. Generally speaking, there should be no concentration within a particular small set of k-mers, or if you were to actually perform an alignment back, you would see they all align to the same area. So Kraken Unique, developed by Florian Brichtwieser, was an attempt to use the hyperlog data sketch and give some estimate of the effective cardinality of the set of k-mers that were used to classify a particular species as being present in the sample. So in the example that is shown here, they've got this C. difficile classification where 122 reads were classified, but only 112 particular k-mers actually ended up supporting that. So you would generally expect this to be significantly higher than that, or in the case that we see here, this is only a small number of k-mers, but this is also a very small genome, as viral genomes tend to be. So it's much less of a concern. So the results of Kraken Unique are really helpful for eliminating certain false positives, a certain class of false positives that tend to occur. And for that reason, especially if you are really concerned with diagnostics, looking into using that can be of considerable value. In Kraken 2, we've actually introduced this HLL and its ideas back in version 2.1.0. You can just run run Kraken 2 with the report minimizer data flag, and it will tell you another column with all of the distinct minimizer counts associated with each thing. We've also published a protocol paper in Nature Protocols last year, where the use of this flag is described as the Kraken 2 Unique program. So you can look through that for examples on how to specifically use it and improve your results in that way. So, I guess in summary, the traditional alignment approach, even though it is sort of the easy thing that we might want to use, and it certainly makes sense, for this problem of taxonomic labeling, it ends up being considerably more work than we might actually want to use. And so some other method, I'm very, somewhat fond of Kraken, but there are other ones out there. Some method that is specifically tailored to doing this taxonomic labeling will still give you accurate labels, but will give it to you much, much faster. And we continue to make some improvements on that through the second major version, where we were actually able to improve accuracy, speed, and memory efficiency for the users out there. I need to thank a number of people who either helped in terms of reviewing manuscripts or contributing code, or just giving us good ideas along the way. So I'm very indebted to all of them. And I also want to thank all of you for inviting me to talk with you today. And I'm happy to answer any questions that you have.
Sure. Marcelo, allow me to ask Derrick a first, not too technical question. Are you a fan of Pandora? What's the idea of the name? How did you come with the Unleash the Kraken?
Oh gosh, the Kraken. It's strange. I did not anticipate getting, when I named it Kraken, I did not anticipate getting the question of why did I choose the name. And yet I have got, it's consistent over the years. Everybody wants to know. So largely, so Kraken 1 actually used a program called Jellyfish to build its thing. So I was kind of going along with the marine life thing. Wanted to call it Octopus initially. And then as I was searching images, I saw a Kraken, and I said, well, that's much better. You got Kamers, you've got, I also like the idea of just using a cryptid as the name for something that's searching it in the dark for random species. So I like that.
Sure. In this series, we learned the reason for the name of salmon. The guy also likes fish and marine stuff. And also Cromwell, the pig, and etc. So it was, I had to ask you.
Sure.
Marcelo, do you want to? I have many questions, but I'll ask after the students.
Yes, my microphone was off here. Derrick, thank you very much for your talk. It was really impressive. It's not a question, but Kraken Unique was a problem solved to us here at our lab. We're going to present next week one poster at the conference. Is called the Bioinformatics, CSI Bioinformatics. It's how I discover my genome was wrong, or something like that. And Kraken, it's a long story. We will take this for another time. Thank you. We have a few questions here that was posed by students. And the very first one is from Vinicius that I lost here. Where is it? Right here. Is here. I work developing pipelines for virus identification in RNA-seq and metagenomics data. I've seen Kraken used multiple times in studies to determine the entire microbiome. But when it comes to viruses, there is a preference for diamond. Why Kraken is not a top choice for separating viruses from other organisms in microbiome?
So I can only guess to that. That's not an area I specifically work in, but I suspect it actually has to do with some of the results we see here. Now, we didn't compare directly against diamond, but I think it is the fact that the translated search that I know diamond does is very likely to give you improved accuracy over nucleotide search. And I think that Kraken is much more known as being a nucleotide search program. That's typically I guess it's typical use case, but I think the translated search should work very well. So I can't answer the question exactly, but I suspect it has to do with the fact that the translated search ones are generally going to be better for viral investigations.
Okay. Interesting. Very interesting.
I was curious, Marcelo and Derrick, if it would be okay, if it would be better to to filter off conserved genes like ribosomal RNA and very conserved ones. Do you think it would help at least in the execution to do that, to filter off some regions that will be very conservative and too much conservative, like the ancestor of those would be in a very root place?
My intuition on that is that I don't think it would have a strong impact
on the result. And
right.
even in the processing, you should not pay too much to process them together.
Yeah, I mean, so the k-mers associated with those regions then would be much higher in the taxonomy. But I think one thing, I have not looked at this specifically, so I don't know for sure. If the level of conservation is not so high that the 31 mers are exactly the same, if there's even a slight differences in there, then there's no need to mask them out, because actually there's informative differences present there. So yeah, I think
not important.
I think so.
Ribosomal is not transcriptomics that the most expressed conserved ones would be the major part of your sample. So I don't know, ribosomal RNA in bacteria is like what, 1% of the genome? It doesn't matter, right?
It's been years since I worked with bacterial transcriptomics, but I know certainly one of the things we did encounter, though, is you're very right about the percentage of the genome that's covered by the rRNA. However, what actually gets transcribed, depending on, what gets transcribed and what gets sequenced, depending on what reduction steps are taken, can actually be very concentrated in terms of ribosomal RNA.
But metatranscriptomics is not a problem. You can run Kraken too in metatranscriptomics if you have the sample, right?
Sure.
Some people ask it also in YouTube.
Yeah, this is a question on the YouTube, and personal experience, we use it and it worked really well.
Oh, great.
More questions from YouTube. You're
yeah, we have another question. Yeah, we have a lot of questions, and fortunately, some questions are arriving through my personal WhatsApp here, and there's some questions. It's one of my questions as well. It's today you have in the market two great players on this transcriptomics, metagenomics, or taxonomic identification. Is Chime, Kimi, or Kime, or whatever, and Kraken. What is the main difference of them? Where Kraken is better than Chime on?
Oh gosh.
Actually, it's not fair to compare because it's another type of sequencing.
It's just another type of. The two questions are here. It's going in this sense. Which one the best for taxonomic classification for metagenomics, Kraken or Chime 2? This is a really tough question. It's hard to compare both of them. But well, it's on you.
Oh yeah.
Someone has to run shotgun and 16S amplification in the same sample, right?
Yeah.
Yeah. So I will say, you know, Kraken 2 does work with 16S stuff itself. In fact, we've got support for using some of the more common 16S databases out there. And so, in fact, I know Jin Liu, I believe it was in 2020, published a paper focusing on using 16S and comparing Kraken and Chime together. It's been a while since I looked at it, but I believe the results of that were that Kraken 2 performed better than Chime in that analysis. So yeah, I think everybody should use Kraken. Why not?
Yes.
Derrick, let me redo the question in this way. Which one is less expensive? So actually the question is, how down can we go on coverage to be in a good place with Kraken? Like, I'm not going to do shotgun to analyze pathways and etc. So I can shut down, I can sequence less. Did you play with the amount of sequence and diminishing the amount to see if Kraken is with the same performance?
I have not looked at that specifically, but I don't think I don't believe that would be an issue for Kraken. That's you know, Kraken.
Sure. That would make a lot of sense for the guys that are asking, because they are going for Chimney because it's less expensive, because they're amplifying sequence. But maybe Kraken could solve the problem easily without the PCRs, and but with just a few sequencing, right? You know, that's why I'm asking if you could cut down and see the performance.
Yeah, so I will say there's fundamentally nothing prohibiting Kraken from working on a smaller reference database, and if doing that, you know, provides somebody with the ability to operate more cheaply in the lab.
If it delivers the same results as Chimney, I would prefer to do a small shotgun and Kraken too, than do the PCR.
Yeah, I mean, I from the computational side of it, I have no preference one way or the other if you were to go shotgun or amplification.
No, I know, but you do not have the data. You did not do the experiment, starting with the samples that you have there and diminishing, calculating the cost to see if you still have the same distribution, cutting down the number of reads. You do not project the number of reads. The number of reads is what people do for shotgun, right?
Right. Yeah, I wish I had known I'd get this question, or I would have taken another look at the 16S Kraken 2 paper before we talked. But I think there's a lot in that paper, from what I remember, that's actually very relevant to this. And I'd encourage the questioners to take a look at that.
More questions, Marcelo?
Yes, I'm reading all the questions here. Thank you, Derrick. It's the power of Kraken is amazing. And one question that is grouping a lot of questions here. Now I'm going to split this group in two questions. I don't know how long can we proceed with this, but I think there is just two more questions here. And this is one very interesting, this group now of questions. One come from Rodrigo Cato. What is the standard size used for K-mer? 23, 31? It gives us a good result using the default, or do you advise to adjust this value depending on the organism?
Okay. I don't. I think that the default parameters should work fairly well for most circumstances. In fact, I don't remember which table it is, but within our publication, there's a supplemental table where we actually did a parameter sweep with numerous different values of K and L, and so you can kind of take a look at that for some guidance if you're a little curious as to how varying those might affect your particular thing. I don't believe that you will get substantial improvement in accuracy by reducing K. You may get increased speed or reduced memory by doing that. So that may be valuable for you. And depending on what you are doing, that might be fine. But I think that the question is very dependent on your specific use case. So yeah, I think that said, I would tend to default to, or tend to fall back to the defaults that are there. But certainly if you find, oh, I need it to go even faster, or I'm willing to trade a little bit of accuracy to get that speed or reduce memory usage, I think the parameter sweep that's present there can give people a good bit of guidance into how that would work. So I hope that provides some.
Yeah, I'm gonna give you my testimonial. I never change on Kraken the default, and it's okay. It's even when our data set that we know what's in there, it's always the same speed, amount of memory, and everything. It works fine. And
sure.
Yes, sorry, continue.
No, so I think you know, if you go into the parameter sweep, there are actually I think you can get it to go almost like 50% faster by changing some parameters, but there's like very tiny changes in accuracy then. So, you know, we tried to come up with a setting which apparently in your case, Marcelo, has worked out very well, and we hope it works for the general audience. But you know, we're also aware that there are people that are going to want specific tuning of things to their own thing. So those parameters are opened up to people to adjust as they might need.
To play with. Right. It's a playground to for change. Continue on this line. I think this is the last one. The question we have here is from Nico. Oh, my glass is terrible. Yes, it's from Nico. And he starts, thanks for the talk. I wonder if you have, sorry. For some reason, it disappeared from my...
Joel has a question that I'm going to...
Daniel Moreira has one question. Thank you for the talk. I noticed in your recent publication in Nature Protocols that you increased the parameter minimum heat groups to 3. This adjustment aimed to reduce false positive assignments. Could you provide any recommendations on utilizing confidence score or distinct minimizer count information or minimum number of assigned reads to enhance the overall confidence in the final classification?
Yeah, so I think yeah, definitely within the protocols there, we were looking specifically at the diagnostic space where having false positives lowered was even more important than the general case. In terms of those various things like confidence scores and that, I've hesitated to get too deep into that because I think it ends up being very dependent on what an individual needs from their results. So some people, have done their evaluations and said, oh, 50% works really well for what I'm looking for, and it reduces the false positive rates to a level that's good for what I need. Other people using the default of 0 are totally fine with what they're getting there. So, definitely as you increase the confidence threshold, that, does reduce the false positive rate. I think even, something minimal around like 10 or 20% is probably sufficient for most people, but once again, it's very dependent on what that particular researcher needs from what they're doing. The distinct minimizer count information, I have to apologize because I actually am not very familiar with the specific statistics that were reported here in Florian's paper. But the general idea, I believe, is somewhat based off of a low coverage value as well as possibly a low K-mer count. So it's a very good question, and you know what, I need to think about it a little bit more. If that person could actually email me, I would love to try to give a more fulsome response on that.
I have a question about ties. When you have a tie, you have a classification of 2 distinct species. Do you report both? No.
In fact, let's see. So in the case where we have 2 these root to leaf paths that actually are tied, we report the lowest common ancestor of those 2. So it's always one thing gets reported out for a read.
Okay, you don't report the other ones. Okay, great. Understood.
Yeah, we have one comment here from Pablo Peraza. Hello, and thank you, Professor, for your presentation. I'm from Uruguay and now working with GBS data from ruminal content and Kraken too. Seems amazing so far. Insanely faster than BLAST data too. And I'm work with Calm Runner version 1, 19 gigabyte setup for Kraken. Congratulations. Send you a warm thank you. And I think it's the last question we have here from Joel.
There is one more here.
And Joel Nunes.
Yeah, you are going to read this one. Let me do it.
Okay, you can go.
He asks, what database does Kraken use to make its classifications? I suppose that you use NCBI taxonomy.
Yeah, so by default, we use the NCBI RefSeq data from, I believe, archaea, bacteria, viruses, and the human genome, as well as.
But the taxonomic classification, you import the NCBI taxonomy one that goes together with those ones. I would like to comment, Derrick, that we have a contribution to NCBI taxonomy classification database where we add taxon names for some clades that are missing names there. For instance, if you look for the class for a turtle or an alligator, there is no class there. So we have an algorithm that creates a proposal for that thing. And also for some archaeas, or it depends on the clade, like superorder, subclass, things like that are missing a lot, even in prokaryotic organisms. So if you use this contribution of ours that add names, it's called taxonomy, because all clades have names. I want to understand the impact that I measured that for mega analysis, Megan. Whenever you want to summarize, like how many families, how many classes I have in my sample. Sometimes if you do not have names, you are going to join all of them together and say other ones or no names. But with the names, you can distinguish it, because this class is father of this family, and the other one is father of the other family. So it has a distinct name. Is there an impact for the work with Kraken 2? Is it common to summarize like that? How many classes, how many families we have? Or you just don't do that?
I don't know. I mean, I think so. Questions of diversity type things. I know some people are definitely do some things like that. And I think Jin Liu has actually developed some things to help calculate those a little bit more easily. I will say though, as far as Kraken itself knows, it doesn't, for the most part, doesn't really know what the names of these nodes are. It simply uses taxonomic IDs. So as long as the structure of the tree is good, it can differentiate between these things.
Between samples, but you do not summarize. But maybe even it's a small difference, but there will be some nodes that are created by the algorithm to justify, like I have to have an order for this organism. I place the node over there, and this distinguishes from the other orders.
Sure,
but it's
yeah.
not, yeah.
Yeah, so I think in that sort of case, what you would be doing is, I mean, you'd have to create your own taxonomy prior to building a Kraken 2 database, but you could do something like various OTUs based off of whatever algorithm works best for you, as long as the maybe going back a few slides here. All Kraken really cares about is it's got a set of genomes, a taxonomy, and some mapping. So you can specify a taxonomy that you feel makes the most sense for you, as long as this mapping is correct within the context of that taxonomy. Then it can perform those classifications as best as it can there.
Your taxonomy is loaded in the memory as a tree, but that does not use that much memory, or do you think so? No,
it's very, very minimal.
Minimal, very minimal, yeah. Because actually, when you have names for all the clades, you can work with the taxonomy exactly as you work, but as a table. The common ancestor is in a point in a table, because all the clades, all these species have the same number of classifications, But maybe it's not that useful.
Yeah, I mean, so definitely for some downstream analysis, that's a thing people definitely want to take a look at. The names end up being much more valuable to humans than the tax IDs. But within the actual program of Kraken, especially within Kraken 2, it's just a pretty simple. Actually, it's not even a tree technically; it's just an array. But it's only maybe a couple of megabytes. It's not very big. Problem.
I have two more technical ones. Do you think that's possible to work only with A and T's, just to work with transitions and not with transversions and not transitions. That would be more rare, more representative. Would it help to filter off C and G and use A and T only? Would perform maybe with a longer camera. I don't know. Well, if you have to have double, you have to double the size of the camera. It doesn't work. But maybe just to avoid the transitions, you know, A to G, C to T. Maybe you could have more rare events.
I don't know. That I'd have to really give that some thought in terms of how we would implement it.
Sure.
Yeah, I think.
Good.
I mean, primarily the sort of underlying idea of Kraken is just an alignment based thing. So we'd have to see
sure.
how much difference we would expect to see there.
Yeah.
Or,
my idea is to mask transitions that are more frequent than transversions. So then maybe should diminish the number of data that you have to look at. Another question related to that, we have a collaborator here in my PhD program that does pretty much the same comparison in the vectorial space, coding the k-mer to a vector and then comparing the vector space. Do you think it's worth asking him to try to develop something with DNA and the size of k-mers that you use to see how it goes?
Yeah, I mean,
would you look at the experiment? Because I think that the vector space is very easy to code, and I think that it reduces the memory and speeds the search. But I don't know if it's possible. Okay. But I think that they do that. I will suggest them, and I have your email, so if we succeed a little bit with some examples, we can try to contact you, right?
Sure.
Could be an idea. Okay. I made like most of my questions. Yeah. Marcelo, what do you think? Time to go having beer? You are 1 year earlier there, so
yeah.
Did you have lunch, Derrick?
A little bit, not a big one.
Yeah, for here, no lunch yet, so not a problem. Oh, wow. Well, I think we are run of questions, and thank you very much, Derrick. My invitation for you for the next X meeting is up. Let's talk where we're going to be the next X meeting. The idea of Miguel to be by the beach, it's it's going to be interesting. It's a very interesting option, but
could be Rio de Janeiro. The tickets to there is not expensive. Carol can find a nice place even in Rio.
Yeah, it's one thing to say. So thank you, Derrick. Thank you very much, Miguel. Thank you very much for this opportunity for us to be here on the Brazil webinar. And see all of you in next meeting next week.
Sure.
Thank you very much.
Thanks a lot for coming. It was amazing. It was amazing. We had over 100 participants in our auditorium in UP and in the Zoom. So it was a great success. Thanks a lot. Thanks a lot for accepting. It was a great talk, a great afternoon here in Brazil. Thank you, Derrick.
Thank you for having me. I appreciate it.
Thank you very much. Cheers, guys. Bye-bye.
Yeah.
Okay. Bye.
Bye-bye, people.
People are now living.